Hello everybody, welcome to Voices of Friedreich's Ataxia. Our goal is to raise awareness and funds to cure Friedreich's Ataxia, FA. Today we will talk about resveratrol. Resveratrol is a naturally occurring compound found in the skin of red grapes. Resveratrol has been under investigation as a compound that could improve mitochondrial function, and some studies suggest increased longevity, lowering glucose levels and anti-cancer activity. Researchers in Australia found that resveratrol also increased frataxin levels in laboratory studies. Investigators at MCRI in Melbourne, Australia are continuing to evaluate the therapeutic potential of resveratrol in clinical trials. Jupiter Orphan Therapeutics is a company developing a novel formulation of resveratrol to improve the absorption and pharmacokinetic properties. This information was taken from FARA Australia with their permission. Their link is provided in description. Hello everybody, welcome to Voices of Friedreich's Ataxia. Our goal is to raise awareness and funds to cure Friedreich's Ataxia, FA. Today we will talk about DT216, gene tag. Design Therapeutics is advancing a novel approach to activate transcription of the FXN gene and restore production of frataxin protein. This approach is based on work from Dr. Asim Ansari's lab with development of compounds called synthetic transcription elongation factors. In 2019, FARA awarded the Kyle Bryant Translational Research Award to Design Therapeutics for their research proposal advancing SynTEFs as a therapeutic candidate for FA. This grant award allowed for some of the early testing in animal models. Design Therapeutics built on this early work to develop a next-generation molecule they call gene-targeting chimera or gene tag. This information was taken from FARA Australia with their permission. Their link is provided in description. Hello everybody, welcome to Voices of Friedreich's Ataxia. Our goal is to raise awareness and funds to cure Friedreich's Ataxia, FA. Today we will talk about oligonucleotides. Nucleic acids are the building blocks of DNA and RNA. Oligonucleotides are chemically synthesized short DNA or RNA molecules that can be designed and used as drugs when targeting specific regions of DNA or RNA to regulate or control transcription or translation of DNA or mRNA. Antisense oligonucleotides, ASOs, are single strands of DNA or RNA that are complementary to a chosen sequence and often used to prevent translation of an mRNA into protein. However, ASOs can also be designed to specific targets that can increase expression of a gene. Friedreich's Ataxia Research Alliance, FARA, is funding research in Dr. David Corey's lab at UT Southwestern, where he has been testing ASOs and other oligonucleotides called gapmers that target the GAA repeat in the frataxin gene or mRNA as a way to activate or produce more frataxin protein. FARA is also funding research in Dr. Jonathan Watt's lab at University of Massachusetts, where they are improving the chemistry of oligonucleotides so that are able to reach more specific regions in the brain and the periphery, e.g., heart. In addition, the Watt's lab is trying to identify novel, non-GAA repeat, targets within the frataxin gene where oligonucleotides could be designed to activate frataxin expression. This information was taken from FARA Australia with their permission. Their link is provided in description. Hello everybody, welcome to Voices of Friedreich's Ataxia. Our goal is to raise awareness and funds to cure Friedreich's Ataxia, FA. Today we will talk about Gene replacement and editing. Significant advances in gene replacement therapy and ability to edit genes have garnered much interest as a treatment approach for FA. There are several characteristics of FA that make it a favorable candidate for gene replacement therapy or gene editing approaches. 95% of individuals with FA have the same single gene mutation, GAA repeat expansion. Reducing the number of expansions by gene editing may result in normal expression of the gene. The mutation in the gene leads to gene silencing and thus a reduction of the frataxin protein, thus there are not abnormal or toxic proteins to remove. Individuals with FA do make some frataxin protein, just not enough, and therefore it is less likely that they will have an immune response to replacing frataxin. Individuals who are carriers for FA, one abnormal copy of the gene, only make 50% of normal frataxin and have no symptoms, so even modest increases in frataxin are anticipated to be therapeutic. Newer generations of viral vectors, adeno-associated viruses, have improved safety profiles and have favorable affinity for the tissues most affected in FA, brain, spinal cord, heart, muscle. This information was taken from FARA Australia with their permission. Their link is provided in description. Hello everybody, welcome to Voices of Friedreich's Ataxia. Our goal is to raise awareness and funds to cure Friedreich's Ataxia, FA. Today we will talk about CTI-1601, frataxin. Dr. Mark Payne, who first described the possibility of frataxin replacement therapy via frataxin, has been developing this exciting prospect for FA therapy. Synthetic frataxin protein is partnered with a unique delivery system, a protein fragment called a transactivator of transcription or TAT, to get the frataxin protein to the mitochondria. Dr. Payne has tested TAT frataxin in the FA mice and demonstrated proof of principle and compelling results. See publication below. Dr. Payne's approach increases the lifespan and weight of FA mice and improves their cardiac function. Dr. Payne formed a company, Chondrial Therapeutics, to further develop this approach and generate preclinical data. Chondrial is now Larimar Therapeutics and clinical trials of CTI-1601 are in progress. This information was taken from FARA Australia with their permission. Their link is provided in description. Hello everybody, welcome to Voices of Friedreich's Ataxia. Our goal is to raise awareness and funds to cure Friedreich's Ataxia, FA. Today we will talk about etravirine. Etravirine, brand name, Intelance. Dr. Roberto Testi and his team at the University of Rome and Fratagene recently reported results of a drug screening study. They developed a cell-based assay to conduct a high-throughput screening of 853 FDA-approved drugs in search of drugs that could increase frataxin protein levels in the assay. Among the potentially interesting drugs identified as apparently able to increase frataxin protein levels in the cell-based assay, the team focused on etravirine for the next step. The team then introduced the drug to some fibroblasts, skin cells, and lymphoblasts, blood cells, derived from FA patients. In those cells, it seemed able to increase frataxin protein levels to some extent and restore activity of an iron-sulfur cluster-containing enzyme, aconitase, that is decreased with frataxin deficiency. Etravirine appeared also able to protect FA patient-derived cells from oxidative stress. Initial studies to clarify the mechanism of action also indicated that etravirine might be able to increase the translation of frataxin messenger RNA, mRNA, into frataxin protein. The team reported to the FA patient community at a recent meeting but has not yet published the fact that it then gave the drug orally to one of FA mouse models. In those mice, the drug appeared to increase frataxin protein levels to some extent in skeletal muscle and heart. This information was taken from FARA Australia with their permission. Their link is provided in description.